From MicrobeTV, this is Office Hours for Wednesday, July 17th, 2024. I'm Vincent Racaniello, and welcome to our little corner of the internet where you can learn good science. I'm in New Jersey tonight where it is currently 28 Celsius and cloudy. Joining me tonight from Tacoma, Washington, Mark Martin. Welcome back.
It's good to see you, Vincent.
You were last year in January. I checked. I think every 6 months is a good rotation schedule. But then people forget about you, right? Well, as Peter Palese used to say, you never have to worry about repeating things because no one listens to begin with.
Yeah,
All right, everyone. Go ahead.
all I was going to say is that's the advantage I find in my personal life is that I talk, as my wife says, I talk a lot of shit. So I repeat myself a lot. So eventually it all gets in there.
No, I think it's okay to repeat.
Welcome,
everybody. Welcome, and let's thank our moderators tonight. We have Les. We have Andrew from New Zealand. Les is from California. We have Steph from San Francisco. Barb Mack from the UK. And Tom Steinberg, who is in the Oregon Coast Range tonight. Thank you, moderators, for being here and ensuring civility. And thanks to all of you for coming back. I'm going to have you tell us where you're from in a few moments. But before I do that, let's see. A couple of things. The agenda for tonight, I have a couple of things to show you. We're going to find out where people are from. And then I have some things to talk with Mark about. And you can always ask questions at any time about anything. And if we don't know anything about it, we will tell you. I wanted to show you a few things that people have sent me over the past few weeks. The first is a little wooden plaque, which says TWIV, right? It's carved. And it is made by Jeff Smith. Jeff Smith is a virologist in at the University of Oxford in the UK, Professor Jeffrey Smith. And he carved this and he sent it. Did he put a note in here? Let me see if I have a note in the envelope. No, there's no note. But Jeff carved it. And Jeff is a well-known pox virologist. And he's not on the stream, but he does listen to TWIV. And we've often covered his work. And if you've ever seen the video of a plaque formation, a microscopic plaque forming, he's the one who did that. It's one, in my opinion, the greatest video, the greatest movie of all time. So thank you, Jeff. He even carved his initials in the back. Jeffrey L. Smith, a lovely virologist. So thank you very much for that. Okay, next, Jessica. Jessica from Toledo, Ohio, who is often here, gave me a nice package. So he has a little card here. Thanks for being you. I don't have any choice in the matter, right? So you're welcome. And then she sent a bag of chocolates, which I left at the incubator because that's where people should consume chocolates, and a pair of socks from the University of Toledo, College of Medicine and Life Sciences. Look at that. And she wrote in the card, go Rockets. I guess that's the nickname for the university. Look at that. There's a little staff, School of Medicine. These are cool socks. I like them. Thank you, Jessica, for that. And Jessica may be on the live stream tonight. She often comes. And one more thing. A listener who asked not to be named, hand knitted 16 caps, different colors, and they have DNA on them. Because as you know, DNA is everywhere. So here's a red one, obviously. And I really like this one, this white one. Wow. Isn't that cool? And
those are beautiful.
We are going to devise some kind of a contest to give these away. Crooked. Look at that. Don't I look 10 years younger automatically?
Yes. That's a hipster look.
I love it. It's kind of a short cap look, right? It doesn't come all the way
Yeah.
down. But I
Yeah.
love the DNA, right? She even put the bases in there. Look. So we're going to devise some kind of a contest to give these away to listeners. We haven't come up. Jessica's here. Thank you, Jessica. Actually wrote to you on LinkedIn, Jessica, and I said, was this you who sent this stuff? Because I want to thank you. So thank you very much. And these hats, we're going to come up with a contest, and maybe Mark can advise me of cool things to do, because Mark likes to give things away too. Okay. Is there anything else? No,
I do.
that's it. So anyway, when you send me things, I will show them here on the program, because I like to thank people who give us stuff. Can you bid for them? Oh, that's an interesting question. Bidding would raise money directly for MicrobeTV, right? And if you really want, so there's white, or kind of off white, there's red, there's brown, there's yellow, there's one yellow one, because the knitter used up extra yarn that was sitting around. And it mentions that it's all natural fibers, no synthetic stuff. It's really cool. I like knitted hats in the wintertime. Maybe bidding is a good idea. What do you
Mm-hmm.
think, Mark?
I think that that's one way to raise money for MicrobeTV.
Hats must go to microbiology students, surely. Well, you could bid it for your own use, or you could bid it and donate it, right, to a
Yeah.
microbiology student. But yeah, bidding is a good idea. We'll make a list of ideas and think about them. But hopefully, maybe towards the winter, towards the cooler months, we will do a contest of some sort. If not bidding, then something else. You know, um, we did the contest once during the pandemic. We had some textbooks to give away, Principles of Virology. We had people write poems. Oh my gosh, we, we had the most amazing poems
Mm-hmm.
on the planet. It was really hard to rank them, but we ranked the top 5 and gave them textbooks. We read them all on a couple of different TWIV episodes. Very talented people listening to us. So, thanks you guys for sending that stuff in. I really appreciate it. All right, let's see where people are from. I bet you're the only person from Tacoma, Washington tonight.
Probably.
Let's see if we can prove you wrong. Okay, Dawn is from the Netherlands. I recognized Dawn for a long time this year, for a long time, many years. And at the beginning, before the show, we were talking about fear and ignorance, and we'll get back to that later. Here is Jessica from Toledo, Ohio. Thank you. And Jessica gave me the chocolates and the sock and the nice card. Okay. Okay. Nice sock. I like socks. I like hats. I like glasses. Look at my glasses collection. I got green. Of course, I'm wearing red. I have green. These are the blue ones I used to wear. I have a light blue one. These are all $2 glasses from Amazon. I have a dark blue one. I have a brown one, if I want to be serious. And these are the glasses I wear on a daily basis, right? I like those. Those are cool. And I have, these are interesting red ones. They're a little small for me, but I kind of like the look. And finally, remember Iris Idris, I think her name was. She just died at 102. She used to wear big glasses like this. These are like architect's glasses. They don't really fit me, but they're fun. I have lots of glasses. Do I have any more? Here's a case. Nope, it's empty. And I have just as many at the incubator. Anyway, thank you, Jessica from Toledo. Andrew's from New Zealand, of course. Jan is from Tucson. Mr. Doreen is from
Elgin.
and not 35 Elgin. Okay, sunny and not 35, so it's a little cooler today. Barb Mack is from the UK, of course. Elizabeth is in West Virginia, 26C and breezy. Lise is from Columbus, where the weather is great. As I said, Steph is from San Francisco. Kang is from Chicago. He says, we need more passionate people like you to rouse people from the sleep of reason that produces monstrous fears.
Mm.
Well, Mark is one of that persons also, and he now has a platform on MicrobeTV which he can use, and he is very good at using it. He's owned it, so that's the thing, Mark. You own Matters Microbial, because I started all the other podcasts, with the exception of Infectious Disease Podcast, which is definitely Daniel's. The other people don't want to step up and own them. I think the closest one is TWIM. Michael Schmidt could own TWIM, right? I don't need to own them forever. That's the thing. I feel I got them going. I need to own TWIV forever. That's the only one because it's the virus one. But the other
Sure.
people could step up and say, I'm ready to own this. I'm ready to take care of everything that has to be, because there's a lot behind the scenes, Mark. Okay. Did you know? So Mark owns Matters Microbial. He has the direction, he has the vision, he invites people, he schedules it. That's what I'm talking about, owning it. So I don't have to do all that. And if we're going to grow, one person can't do all that anyway. So, yes, I would, and I want, if you own a podcast, and the word own doesn't mean possess, it means you run it. If you own a podcast, then you can be a passionate person who rouses people from the sleep of reason that produces monstrous fears. So I guess, Mark, I'm trying to train people to do this, right? Sound reasonable?
It's been a dream of mine to be able to talk about my passions for the sciences, and this is a good way. I mean, tomorrow I record my 50th episode. Unbelievable. And we're talking to Will Ratcliffe on the evolution of multicellularity, which is just fabulous stuff. I'm really excited about it.
You need to get Nicole Smith. Is that her name? We talked to her on TWIM. Let me find Nicole. She also does multicellularity. Nicole multicellularity. What's her name? Nicole
Oh,
King
is
at
this?
Berkeley. Nicole King
Yeah,
at Berkeley. Okay.
these are the choanoflagellates. Yeah, they're fascinating.
Choanoflagellates. Right.
Mm-hmm.
Anyway, thank you, Kang. Are there more microbes than us in us?
Now that's an interesting topic.
They used to
And,
think so,
and
right? And then they screwed up the math, right?
I really love this story. I tell students this a lot whenever they read things. Turns out that when you go, I'm going to call it reference spelunking, when you go chasing down references, and the prediction of how many microbes exist within us pretty much based on the gut. And this is because it's so incredibly dense with microbes. But it turns out that the initial guesses, and it was originally 10 microbial cells for each one of ours.
Mm-hmm.
Turns out that original guess from, I think, the 70s was just that, a guess. It was not backed up by anything. Then everyone started quoting one another, you know, and it turns out it's about 3-1, the last I heard. And to be delicate, when we eliminate some of our nutritional insides, the number comes a little closer to 2-1.
Mm.
Was that graphic enough for you, Vincent?
2-1 bacteria to human cells.
Yeah. So in other words, when you eliminate some of the
Yep.
insides of that. So we're tubes. That's what it is.
So there are more microbes than us in us still,
Yeah.
right? Okay, John. It's a good question. So we're going to talk about this in a bit, the latest with RFK Jr., but Kang said, FDR said, fear is a nameless, unreasoning, unjustified terror which paralyzes needed efforts to convert retreat into advance.
Yes.
That's true. And people are afraid of many things. They're afraid of vaccines, apparently, and they shouldn't be. And there's a decreasing trust in science and scientists. They don't want to believe us, and they would rather believe politicians. And we'll talk about that in a moment. It's really a scary thing here. Okay, let's see. John is in Minneapolis, Minnesota. We're at 73F. Thanks for coming back, John. Here's Tom in the Oregon Coast Range. Abdul Aziz is in Saudi Arabia, I'm pretty sure. Pete is in London, low 20, 16C at 1 a.m. Thank you for coming around at such a late hour. I know what late hours are. I was out at 1:30 this morning, Mark. I did a live podcast in Australia. Oh my gosh. 1:30 in the morning. I stayed up. I did it. Dagfinn is in Oslo, Norway, 15C. It's summer. Oh my gosh. It's a cold climate for a summer. But, you know, if you look at the latest Instagram posts, Norway is one of those good countries to be in.
Mm-hmm.
You know what some of the top cities are to live in, apparently, like Geneva and Zurich and Copenhagen. You know, I used to say Copenhagen, Mark, and Elio said, Vincent, you don't have to make a German name out of it. I love the way Elio corrects you, right? He just tells you, and he's so nice about it. Even though
Yeah.
his words are cutting, you don't mind, right? Okay, who else is here? MJ is coming from Wilmington, North Carolina, where it's 83F and overcast. Who else is from somewhere? Well, everyone's from somewhere. It's just a matter of telling us. Abdul Rahman is from Colorado Springs, driving to Denver. That's pretty cool. Jonathan from GSD Bio. This is your new friend, Mark. Is from Encinitas. Patty is in Massachusetts. Mike and Lori are in Seattle, not too far from
Yeah.
you, right?
About 35 miles. Yeah.
Oh, that's very close.
Mm-hmm.
That's the distance from where I am right now to Columbia University Medical Center, 38 miles. I used to drive that every day. 35 miles could sometimes take 3 hours. Mm.
Yeah.
Tona is from Long Island. Long Island. You get it, Mark? You get
Long
that accent?
Island. Yeah. Yeah. Long Island.
Long Island. Uh,
Hey, Vincent, I have a new toy for you.
oh, it's a tardigrade.
Yeah.
Come back. Come back into the frame. It's segmented. I hear it clicking. Now, where'd you get that one?
Um, I have an addiction to Etsy, and
Yeah,
that
yeah.
particular model is not expensive. You do have to put it together. It's 3D printed.
Mm.
But you know, I usually have it perching on the boom to my microphone.
You have a lot of stuff behind you.
I do. I
It's
do.
okay. It's your office.
Mm-hmm.
What are you going to do when you vacate your office? What are you going to do with it all?
Um, well, this is a subject that my wife and I have discussed a couple of times because she has to have an off. Well, she's working from home too. And so she is saying she's going to refit that one room into her office. Well, I think Dr. Martin needs an office as well.
Do you have room for an office?
Yeah.
All right. Well, there you go. All your stuff goes in there.
Yeah. The kids are gone. So there you go.
MJ said she's excited to see Dr. Martin. I love Matters Microbial. It's so educational, entertaining, and inspiring. See, Mark?
That's nice. Thank you so much.
There you go. Cheryl is in Santa Rosa, California. That's a very pretty place, isn't it?
It is.
Markle is the east end of Long Island, a gray, ominous day. Upstate had 4 touchdowns, which may be classified as tornadoes, and 1 man was killed. That's unfortunate.
It is.
Curly Dog is in Montreal. Got COVID from his wife. Sorry about that. Privyet Veronika from Moscow. 21C Privyet. I know very few words in Russian, Mark. And I've mainly learned them from here. I know Privyet and Poka, which means goodbye. And I know Spasibo and
Mm-hmm.
Dozavtra, which I think is like
Zavtra.
thank you or something. Dozavtra. So Privyet, Veronika, and thank you for teaching me some of those words. I like to have a few words in some language, right? Who else is here? Yeah, Jessica, nerd swag. Is that what you call it? Nerd swag?
So Vincent, in all my classes, I create nerd swag for
I
everybody.
know you do. Yes.
Yeah.
But
Uh-oh.
look, who do you think gave me this? This 3D printed tardigrade.
Yeah.
Yeah, it's very pretty. I have lots of stuff from Mark. And I have
Uh-oh.
another. Yeah, here's another. One more. Here's another one. Thulu.
There he is.
It's a pretty scary guy, or whatever he is, a thing. Nerd swag. Yeah, you make a lot of cool stuff for your class. I
Right.
always
I
want to.
decided.
I'm
Go ahead.
always inspired by you, but I never do it because all of a sudden it's time to teach and I don't have time to do anything.
No, absolutely. So in the fall, when I'm teaching microbiology, they all get, I make artwork for each year. So I have to finish the artwork for this year, which may be based on the Frank Sinatra thing. It's a microbial world. We just live in it.
Yeah, yes. I
The,
have
the
those.
Dean
I
Martin
have
thing.
those
Yeah.
buttons, man. I have plenty of those buttons. Yeah.
Yeah.
Did you
So it
see what
may
I
be based.
showed last week? You see what I showed last week,
It's
Mark?
beautiful. Yeah, I was
This
there.
is
Isn't that lovely?
gorgeous. This is
Yeah.
so beautiful. I love it. She did a great job on that. Yeah.
Yeah. Yeah. So there, I make
Uh,
T-shirts for the class,
right.
buttons, stickers, that kind of thing.
Do you
What
do
I
it
like,
before
what I
class or do you do it when class starts?
first day of class. What I like to do is give these the giant microbes people, which are moving away from micro other way. Mark, there you go.
Clap the clap.
Yeah,
Gonococcus.
yeah. So then, of course, people say yes. Dr. Martin gave them gonorrhea. That's not funny, but they say it all the time. They find that endlessly funny.
That's great. Elise says, I'm getting a big sock collection. I am. Yeah. Elise gave me a few pairs of socks as well. I like socks. Frank is in Santa Barbara.
Beautiful.
Very nice. And Carol is in Southern California, our 101F. Wow. Our favorite nephrologist. Welcome back, Carol. Nerd swag. Nerd swag is the coolest swag. I mean, all of our TWIV stuff, all of our MicrobeTV stuff, that's all nerd swag, right?
Mm-hmm.
I have all kinds of cool stuff. And I have to start bringing T-shirts again to meetings when I do TWIVs or whatever, because it's always nice at the end to throw them out. People get very excited to pick one. Doreen is in Hartford. Cool being with us all. Thank you very much. Tracy is in Chapel Hill, North Carolina. And Andrew said the hat is a Gene T cozy. That's right. That's right. It's a cozy for my head. Very good, Andrew. I like that. Yeah, the knitting is very nice. Let us know about the contest. We will, of course. You will be among the first to know, of course, because it's live stream is Wednesday, and that happens before Friday, which is TWIV. Lise is already starting the bid at 200 for the red knitted hat. This is great. You may get it at that, I mean, I don't think anyone's going to pay more, but that's a donation to microbe.tv. It's also tax
Yeah.
deductible, right? Because, well, there's this rule about getting something in kind. And I mean, a hat, that's not a $200 hat, I guess. Don't worry about it. Okay. What else do we have here? Richard is from Portland, Oregon. Doreen is attending an open knit group tonight. Ah, you're going to knit something. That's great. Great. And we have some donations. Laura Potter is here. Hello, Kip and Laura. Thank you for your support of science communication. We really appreciate it. Let's see, how many people are here tonight? We have 101 people, 51 likes. Please hit the like button. It's right below the video. Just hit it. It's free. It helps, we think, attract people here. Who knows who would show up and learn good things, right? John, thank you for your support of science communication. Attempting to bid on a hat for the cold Minnesota winters, but I won't hold you to it. We haven't agreed that it's a bidding yet. We've just said that that's a good idea. I mean, it's a great way to make money, but I don't know how to actually conduct the bidding, so I want to find out from people who have done biddings if that's what we do. Barb Mack, thank you. Oh, bid on, oh my gosh, 50 pounds bid on a hat. Now, am I locked into bidding now, Mark? Is that the thing?
I think you started it. So who's going to write all this stuff down? Fortunately, it's kept in the chat,
It's
right?
kept in the chat. It's kept in the chat. Well, if you bid on something, I have to first of all tell you all the different hats that I have. These are only 2 of 16, and then you can pick the one that you want to bid on. But if we end up doing that, and it seems very likely now that we will keep your bids in mind. Thank you so much for bidding already. HAT is in Los Angeles, where it's 26C. Lucy is in Sechelt, BC. So it's out your way, more or less, Mark, right? BC. CYP3 is from Sweden. It's an enzyme in the liver, isn't it? CYP3. Right? Am I wrong? CYP3.
He's looking it up right now.
These are the CYP3 cytochrome P50 family 3 subfamily A, also known as CYP3A, but the guy from Sweden or the gal from Sweden is CYP3. They metabolize drugs. They're very important.
Yes.
And that's why when you take Paxlovid, not only do you get a protease inhibitor, but you get an inhibitor of CYP3A, because CYP3A will turn over the protease inhibitor too quickly. So ritonavir inhibits CYP3A. So don't people want to understand how things work? That's a very what I just said is a very superficial level, but still you understand what's going on, why there are 2 drugs in Paxlovid. I would think you would want to know that, right? You would want to know how things work.
And I want you to remember, Vincent, that for people who say that they don't really want to know a lot about science, those same people will understand very intricate details of things that they do like. So it's not that they can't, it's all about choices.
Krishnan is from Malaysia. Good morning. Yeah, I guess it's morning there. Thanks for listening. And CYP3 is from South Sweden by Helsingborg and Helsingor. What's the rest of it? Helsingor. It's so interesting you should say that, Mark. I mean, CYP3. Because I was in Sweden a few weeks ago, and we drove to a canal where the boats go through and the locks. And I remember driving through Helsingborg and Helsingor. Kind of sounds like something out of Harry Potter, right? Gryffindor, Helsingor, maybe not. Patricia is in DC, where she had a drought. And MJ is a big fan of yours, Mark. And Dr. Martin is amazing. He is Matters Microbial. Well, you created the show, Mark. It's yours. And Barb says, congratulations on 50 episodes. I want to talk to you a little bit about that. We'll come back to it. We got a lot of good questions here. Rip DeComp is from Taos, New Mexico, windy and 60-something. Professor Racaniello, the Elton John of virologists. That's right. I could wear multiple glasses at once.
If you had the little windshield wipers on them, I think Elton John used to have those.
He had windshield wipers?
Yeah. But I think the problem here, Vincent, is that pretty soon people are going to start sending you glasses to wear.
I don't have a problem with that. They can send me glasses.
With lots of rhinestones on them, that wouldn't trouble you?
No, I'll do it. It's fine.
There you go.
I'm happy to be the role that is needed to communicate. Okay, that's the thing. And GSD Bio says maybe 10 to 1 phage to bacteria. Is that true?
Oh, that's a really interesting comment there because I couldn't begin to. Oh boy, it's probably higher than that. Don't you imagine, Vincent?
Yeah, I think it's higher than 10 to 1 because one bacterium will make thousands of phages, right?
Well, certainly between 1 and 200 for the T even types that I'm most familiar with.
Well, you're just being technical, Mark.
Yeah.
Yes, you're right. But no infected bacterium makes thousands of phages.
I don't know. I mean, I don't know enough about like algal viruses, excuse me, algal viruses. They might, because there's enough cytoplasm there that there might be enough of a factory, to
So
make a lot. It's a
the
very
reason
interesting subject.
the reason I ask is because eukaryotic viruses routinely make thousands per infected cell, and I was assuming it was the same, but perhaps not. Right? Doreen says, "Thank you for the late hours you give to these valuable and fun podcasts." I don't think twice. In fact, down in Australia, they said, "Oh, aren't you going to do this twice a year?" And I said, if you wanted me to do it at 1:30 in the morning twice a year, I'd do it. I did not hesitate when they asked me to do it at 1:30 in the morning because it's important, and my desires and needs are subsidiary. No, not what's the word? Sub something. Subsumed by
Subsumed.
the greater goal of what we do. SIP3 says, extremely beautiful little place to explore, Helsingor. I'm going to go back to Sweden many more times in my life. Maybe we can meet up. I have a friend there who is a TWIV listener, and she takes me around. And I showed pictures of these locks a couple of weeks ago, SIP3. It's very cool. So Pete Glass is not going to stay up till 3 a.m., which is when we're done here. He's giving his 50th blood donation tomorrow. Very good. Congratulations. I hope that's been spaced out over like 12 and a half years, maybe. Dagfin says this is unusually cold for Norway in summer. Besides, it's nighttime. Could you have Dr. Condit send some Austin weather our way? Oh my gosh, yes. He would love to. Cool it down. Mr. Doreen says, thank you for countering a miasma of anti-think. That's a lovely saying, which someone came up with during the pandemic. We had actually T-shirts made with that on it. TWIV, countering a miasma of anti-think. It's really good. All right, who else? Hello from Brooklyn, from Lucien Desar, with a temperature, well, you know how hot it is. Face orange, raised eyebrow. Thank you, Lucien, for coming by and from Brooklyn. And Touj 410 said, I had a coworker say that COVID and the flu are the same thing. Well, they're both viruses, right? But that's not the same thing, right, Mark?
No, and I know why this happened. Because people kept talking about all respiratory viruses being the same problem during parts of the year, when of course they're very different things. What is it that your partner often says, that there's nothing to keep you from getting more than one disease at the same time?
Yeah, you can have as many infections as you like. It's true. And we
Yeah.
probably do routinely have multiple infections. We just don't know about it.
Mm-hmm.
All right, so Touj has a good question coming up, but we'll wait. EG is from Mississauga, Ontario, Canada. Very cool. Maui Swift is from Vancouver. Beautiful place. Not too far
It is.
from you, Mark, right?
Mm-hmm.
How many miles?
About 150.
A little bit more than what was the other British Columbia, right?
Well, Vancouver is in British Columbia. BC is a province.
What did you say was 35 miles away?
Seattle.
Seattle, that's right. Okay. Subordinate. Yes, Les just texted me. One of our mods, Les, just texted me the word is subordinate. I am subordinate to my mission, right? I have no problem being subordinate to my mission. Santa Rosa is the home of Snoopy. I didn't know that.
Huh.
Dagfin says, in your ever improving Russian, you should know how to say "pazalusta," which is like an analog to please. Okay, I'm going to write it down on my list. Are you a list maker, Mark?
Yes, but then I forget where I have my lists.
Hey, I do the same thing. I've
So
started to
we need a list for our lists.
PAZHALUSTA, which is like analog to please. Okay, thank you very much, Dagfin. Oh, Jessica met Karen. Yes, Karen is wonderful at ASV. That's right. Dawn is from Regina or Regina. I don't know how you say it. Canada. Is that close to you, Mark?
No.
Okay. Sharp is from Vancouver. The hat is awesome. It's a great hat. I love it. Yeah. Natalia 7 is from the Philippines. Welcome. Sharp says, live stream that knitting. Yeah, that would be cool.
Can you imagine a time lapse of that and how beautiful that would be to watch?
Yeah, that would be very cool. If I were a knitter, I would video my knitting and time lapse it. That would be a cool YouTube channel. I'm sure it's done. I'm sure people have done it. Yeah. CCX is from Galicia, Spain. Welcome. Very cool. I saw a post on Instagram the other day. You know, you got to take Instagram posts with a very large grain of salt often. And this post said Spain is the best place. The Spaniards are the healthiest people. They eat really well. They use olive oil instead of butter. They take naps, and they have strong social networks, which is really important for good health, apparently. Okay, please in Russian uses a Z, which sounds like in English pajalusta. Pajalusta. Okay. What is that one of the words I have? Oh, yeah, pajalusta. Oh, it sounds like an English J. Pajalusta?
Jalusta.
Pajalusta. Do you speak Russian?
No. Or I should say niet.
Yet. Yet. Anyone else? BC Clarity is from Eastern Oregon. Probably not too far from you either, right? It's the adjacent state.
It is, and over on the other side of the mountains, so it's pretty dry.
Doreen says, we do want to know how things work. Thank you, Vincent and Mark.
No, I think, and Vincent and I talked about this a little bit. I think that, and I don't want to take this off track, but I think that people are interested in things if you couch it the right way. And what happens is that when someone oversimplifies, even if you don't really understand the topic, you recognize that they're oversimplifying or hiding something. And that's
Right.
where a lot of this comes from.
Elise says, please notify me if someone goes over my bid. I will raise them. I want that red hat. That's great. This is lovely. Patricia, good evening, everyone. How are you all? Thanks for Principles of Virology. My pleasure. Where are you from, Patricia? Mr. Doreen says that's why we need to check for medication interactions before we catch COVID. Yeah, so the Paxlovid, the ritonavir, if you have drugs, if you're on drugs that are metabolized by CYP3A, you have to make sure before you take the ritonavir because it will change the bloodstream levels of your other medications, which may not be good. That's the bottom line. And you should understand that. Many people don't get it. I mean, I'm a big fan, Mark, of understanding things.
Mm-hmm.
And I think everyone can understand a lot, but they choose not to.
I think you're right. And I've always been interested in so many things. And I wanted to know more. In fact, I would say that that's probably what ought to be on my epitaph. I would like to know more.
I would like to know more.
Monod apparently, his dying words were, I am trying to understand, apparently. But for me, it would be, I want to know more.
When I was in grad school, I used to work very hard, and my friend said, they're going to put on your gravestone, I worked weekends.
Yeah.
It's okay.
You know, we got
Um,
we got into this a little bit, Vincent, with some students because there's this issue going on about work-life balance where people say, you don't want to work 80 hours a week in the lab. And no one told anyone to, well, that's not true. I know people who used to say that, but you know what I'm getting at. But really what you're looking for if you're looking for someone who's a good researcher is someone who wants to know what happens next. They want to go look in the incubator to see what happened. That doesn't mean 80 hours a week. It just means they're invested in what they're doing.
yeah, that's fine. Not everybody has to do that.
That's right.
I'm looking up something. Yeah. Okay. Um, so Andrew says you can't donate blood despite BSC being 50 years ago, but they just lifted that, at least in the U.S. In the U.S. now, you can donate blood if you were living in the U.K. at the time of the BSC outbreak, but maybe not in New Zealand. So Hickham's dictum, I think that's what Daniel says. He said anyone can be infected with as many agents as they want. It's kind of a play on Occam's razor. Was he a physician? No, he wasn't a physician.
No.
No,
William of
and
Ockham was a philosopher. Yeah.
yeah, Daniel says that Occam wasn't a. So with Occam, the simplest explanation, right? Is that the idea? Is usually the best
Hmm.
one. But he
Usually.
would say you're only infected with one thing, and Daniel said, well, Occam wasn't a razor. Hickham's dictum says you can have as many damn infections as you please.
Yeah.
Hmm,
Do you think that has to do with this big drive to simplify things, to think of one thing at a time?
probably, yeah.
Yeah. And that's not how life is, right?
That's a
We often
reductionist
have,
approach, right?
right, right.
Well, that's how we do science sometimes, right? It's because we have to
Hmm.
isolate something and study it because you can't do up until recently you couldn't look at many things at once. Now you can do omics and do many things at once, right? You can do metabolomics and transcriptomics and so forth. But even then, you get a lot of data and you don't know what it means.
That's right.
So apparently there's a water main break in New Jersey. I have to boil June 2024. I didn't boil any water. I'm still here. It doesn't mean anything. Ah, but Hickam was a physician. Hickham's dictum. Who was Hickam? I looked it up. Who was Hickam? Hmm. I don't get a good answer to that. Laura is from Rainbow Lake in Connecticut. Very hot tonight. Welcome.
Mhm.
Karen's playlist is from Maine. Who else is here before we go back to knowledge is my oxygen? I like that very much.
Nice.
Marco studied Russian for 5 years. It's a different alphabet. Pajalusta. Pajalusta. Oh, there you go. Pajalusta. Emphasis on the pa.
Like popcorn. Yeah.
Pajalusta. Popcorn. Apoptosis or apoptosis, Mark?
So Daniel Kleonsky always insisted that it's apoptosis to me and would correct me every time I said it.
I found a. Right. So here we go. I found a. I should really put this in the. Yeah, I'm going to work my magic and put this in the live feed right now. Okay. Because this is from the first paper that ever described programmed cell death, or apoptosis as we call it. And I want to show you what they wrote because it tells you how to pronounce it. Okay? Let's see if I have to I'm going through a little machination here. Just chill. Just hang on. Make available offline. Here we go. I'm going to drop this on the screen. Oh, here we go. Look, it works. We are most grateful to Professor James Cormack of the Department of Greek, University of Aberdeen, for suggesting the term. The word apoptosis is used in Greek to describe the dropping off or falling off of petals from flowers or leaves from trees. To show the derivation clearly, we propose that the stress should be on the penultimate syllable, the second half of the word being pronounced like ptosis with the P silent, which comes from the same root to fall and is already used to describe drooping of the upper eyelid. Apoptosis, folks.
I'm happy to go with that. I would point out, though, that in the UK, things are sometimes pronounced differently than here. Bacteriophage, bacteriophage.
Yeah, I know. I don't know what to do about that in science, right?
Yeah. I'm happy to go with apoptosis.
I mean, I used to say apoptosis. I got excoriated by my textbook colleagues. I was called unsophisticated. I don't know
Why
why
would they?
being because we know each other too well, and you can call each other names when you know someone too well, right?
I guess.
That's the problem. Les says, on my deathbed, I will say, I wish I had spent more time posting my food photos. Great. I don't get many food photos in my feed, maybe because I don't like any, right?
Oh, there you go.
All right, we're doing a gravestone thing. Marv Mack says, mine will say she never had enough cats.
Is that the
Do you?
toxoplasmosis talking?
Yeah, that's right.
I don't
Mark,
know.
do you have something for your gravestone?
Yeah, I thought I would like to know more. That would probably be a good
I
thing.
would like to know more. That's good. I like that. So that's
Mm-hmm.
tonight's meme, folks. What's going to be on your gravestone? I like that very much. Andrew says Occam gets a bad rap. He said, avoid unnecessary entities in an argument, not keep it simple, stupid.
Mm-hmm.
Well, almost always we misinterpret what someone said, right? Because we simplify it. Yes. Okay. Now I'm getting corrected because I said the wrong thing. Yes. James says Occam's razor doesn't say there's only one answer. It just says of a more complicated versus simple, the simpler is more likely to be right. But
Yeah.
when it comes to respiratory disease, you're more likely to have more than one virus than to have one. Bernard is in DC. Welcome, Bernard. I have a lot of questions for you here, Mark. So we'll come back to
Oh,
them.
sure.
We'll come back to them. And then Andrew has entia non sunt multiplicanda praeter
Mm-hmm.
necessitatem. Things should not be multiplied beyond what is required. Pterodactyl, exactly. But helicopter. Go figure the English language. All right, I saw a great Instagram post on the English language where, you know, in front of many words means not, but in some other words it means is. Inflammable means it's flammable. Okay. Pterodactyl. Patricia told us where she is with tongue in cheek. Thank you, Patricia. I guess I could put that into Google and figure it out. It's okay. That's a very clever answer. I like that very much. I think that's it. Rima. Hello, Rima from Iowa. Welcome. That's our last. Where are you from? I wanted to show you two more things before we talk a little bit with Mark about Matters Microbial. The first is you want a bobblehead of me last week. Lise from Columbus, who had this made and sent it to me, and
Oh
it
my
actually
God.
has my name on the bottom. You can see there,
Oh
right?
my God.
Professor Racaniello. She said you can buy some. So she put up the link last week. Maybe she'll put it up again today. I forgot
Oh,
to
that's
mention it last
amazing.
week. So Lise, if
That
you
is
want to
just
put that
amazing.
link in, you're welcome to do it. You can buy that, although I don't know why you'd want one, but in case you do, there you go. And then the other is there is another meeting in Australia coming up called Options 12. So it's the 12th version of this meeting, and this is about the transmission of respiratory viruses. September 29th to October 2nd. It's happening in Brisbane. And, transmission, zoonotic infections, immunology, vaccine development. Now, the reason I tell you this is because myself, Kathy Spindler, and Steph Langle will be there. And I think we may do a TWIV and an immune. Okay, so if you're in
Wonderful.
Australia, you might want to check it out. Brisbane options 12. There you go. Okay. Mark, what's on your vest? That's very cool.
Oh, well, so there used to be a company that called Infectious Wearables, and then they made ties mostly, bow ties, like Daniel sometimes wears. I'll bet many of his are from there. It was a retired dentist who made this business for many years, and so I have ties like that, yes. But when my mother was ill, she had cancer and she was quite ill, and I wanted to take her mind off things, and she loved to sew. And I was at a place I like to when I'm teaching in particular, I like to wear like a sport coat or a blazer, and people made fun of me about it. So I decided, well, they did. I don't know
Sorry.
why it's their business, but that's just what happened.
Yeah.
Anyway, so I said, why don't you make me a vest? Because I always liked vests. And I bought her the material. They were mostly scarves. I'd have to buy like 3. And then she could make a vest out of it. And I have 4 of them. This is the last one she made. And this is, I believe, anthrax. Yeah, there's a biohazard on there too, isn't there? Yeah. So I like them.
Mark, so you need 3 scarves because of your size? Is that the idea?
Well, that's kind of you, but actually because 2 sides to a vest mostly.
You
The
have more
back
than
is not
one.
the same material.
Yeah, but you had more than one made. Is that right?
I have 4 different
Very
vests.
nice. So you said Matters Microbial number 50 is recording tomorrow, correct? So
That is
how's it
correct.
going?
I really love it. Different times of the year, I have more difficulty cajoling people into coming on the show, and this is because there's only limited amounts of time because of when I'm teaching
Mm.
that are available, and then people. I can't apparently find a way to do people very easily in Europe, for example. I would love to have someone from New Zealand on, but that would be very difficult to do. So in any event, I've been having a lot of fun. I like talking to people about microbial sciences. And so I've gotten to know a fair number of people over the years. And so it's my opportunity to have them on, talk a little bit about their route into how they became a scientist, what really motivated them, and something about their research. So tomorrow, episode 50, is Will Ratcliffe at the Georgia Institute of Technology, who has been studying these snowflake yeast, that is to say,
Mhm.
unicellular organisms that have found a way to become multicellular. And he's
Right.
done a lot of deep thinking about the evolution of multicellularity.
I met him when I visited a couple years ago. Yeah. So Mark, is it root or route? Just joking. It's okay. You don't have to answer. You recently did one with Margaret McFull-Nye, right?
Last week.
How did it work out? Okay, because you went to see her actually.
Yeah. The first thing I want everyone to know is that Vincent has all these things down to a science, especially when he does a remote. Dr. Martin does not. Okay. And poor Doc went down there with a couple of cameras, and the folks at MicrobeTV shipped a couple of microphones, and then I got on campus, and the Caltech network wouldn't let me on, and it was like 900 degrees Fahrenheit in there, because it's
Mm.
Southern California in high summer in Pasadena, and but Margaret was great, so we're side by side in that particular
So you
video.
record
So
on your laptop, right?
that's
Okay,
right.
it's okay. Um, do you have any favorites of all the episodes? It's like asking if you have a favorite kid, I guess.
Well, the first episode that you were on is one of my favorites, just because I
You're
was
just
so
saying that because you're here now. Don't say that. It's not
no,
true.
It is true because I was so nervous and you were really good calming me down.
Okay.
So thank you. Um,
No.
I've talked to a lot of people that I know socially that I see at meetings. Dave Baltris spent a lot of time talking about his research with me. I love talking with, I've known Margaret for decades and she's just fabulous and so darn smart. I had a wonderful episode with Rachel Dutton on the microbiology of cheeses. I've really made a conscious effort to try and find some less represented individuals
Hmm.
to come on the program. I had some pretty good luck with that, which I feel really good about because you know how everyone says something is a big tent kind of idea? So maybe we should say microbiology should be a big petri dish.
Mm.
And I don't mean that in a negative way. It's for everyone. And so I'm delighted to use that opportunity. You've had a lot of new professors. That's been wonderful to talk about the kind of things that they face as they set up a lab, for example.
Okay. Well, we're happy that you're doing it, and we look forward to the next 50.
So my wife is taking the thumbnails, Dr. Jennifer Quinn, and making them into a poster of the first 50.
Very cool.
Yeah, I
Excellent.
look forward to that.
You also went to ASM, American Society for Microbiology,
I
Microbe
did.
in Atlanta. How was that?
Um, it was very different because it was done in front of a live audience, and I'm used
Mm.
to doing this.
Yep.
And I'm not used to slouching over in a chair in front of a camera, but Ray Ortega was great. I did 2 of them in 2 days,
You did
you
one
know,
with
and
Michael Schmidt, right?
I did. He was hysterical.
Yeah, Michael's very good. And he talked about copper, right?
He did, and and you know, we got some unusual comments on the YouTube stream about that from people who I don't think believe it. But you know, I don't engage with stuff like that on YouTube anyway.
Mm.
When you can actually do the study and show that there's a destruction of microbes and in fact viruses too on metallic copper, that was the important point. That was a wonderful session.
Mm.
And
And
then
then
and then
you you did a twim with Michael, right? With Arturo Casadeval.
oh, it was just lovely because Arturo has a new book out and he's just a delight to talk with.
Yes, he's great. And he talked all about fungi, right?
Hmm.
Are they going to take over the world, Mark?
Well, I think Arturo would suggest that they have already. uh,
Yes.
I don't know if that's necessarily true, but it is interesting to note that when people study fungi, when they do, which is a relatively small number of people compared to, say, bacterial or viral issues, they mostly focus on things that cause disease. And
Mm.
I wonder what proportion of fungi in nature cause disease. And my guess is it's pretty low.
Well, the fungi have occupied their niche. They're very happy there, I would say, right?
Hmm.
I mean, some of them are huge, right? 1,500-mile long mycelia underground, right? So they don't have
And
to take over more. They're doing very well.
I think they have been through. In fact, Arturo was talking about during one of the great extinctions in the history of our planet, there's a layer like a meter thick of fungal spores, is what he said in the
Of
fossil
course.
record, which to
Go ahead. Go ahead.
I was going to say, which is just amazing. And I know that there were some of those fungal items. I cannot think of the name of them because I didn't prepare for that, but they were like 3 meters tall. It's like a
Mm.
mushroom, 3 meters tall.
Wow.
But as
And
you
that,
say, and it's important, Vincent, is that with fungi in particular, they spread their mycelia everywhere. So when you're eating a mushroom, friends, all you're looking at, that's the fruiting body of the actual fungus.
right.
It's enormous by comparison.
Yeah, it can be thousands of miles, right?
Hmm.
Biggest organisms on Earth.
Very probably.
I mean, we used to think blue whales were the biggest, but they don't stretch 1,500 miles. So, what was I going to say? Candida auris, right? This is a species of fungus that's gone crazy. And as
Hmm.
Michael Schmidt says, if it gets in your hospital, you're never going to get rid of it.
Yes,
What did
that's
what did
exactly right.
Arturo what did Arturo have to say about that?
The concern, Arturo has a philosophy that what protects us from fungal diseases is our elevated body temperature.
Mm-hmm.
And anyone who's kept a cold-blooded kind of creature as a pet or a companion animal, whatever phrase you want, knows that most animals are really susceptible to fungal diseases if they aren't warm-blooded.
Right, right.
But with the changing climate, the concern is maybe we have a situation where this natural, I'm not going to say immunity, but this natural defense that we have of an elevated temperature may not be quite as effective. Also, there's an awful lot of us on the planet right now, which provides quite an incubator for change.
Yep, absolutely. All right. We have questions we'll get to in a minute, but I just wanted to make one more comment. So Monday, Paul Offit published his latest Substack called Oops. Do you read this at all, Mark?
I did.
So apparently a video was leaked of RFK Jr. taking a phone call from Donald Trump. It was leaked and then got out, and of course they pulled it, but it was too late. In this video, Trump calls RFK Jr. and says, I'm really worried about vaccines. We give too many vaccines to kids. We have to stop this. We have to change the way we do this. And he says, we're going to win, so I want you to be part of the administration and deal with this. So this is very scary because here a guy who has no medical training whatsoever, who has never designed a phase 1 safety dose escalation trial, thinks that he needs to change how vaccines are administered. And as you know, in an interview with Lex Friedman, RFK Jr. said he could not think of a single vaccine that has been beneficial. And Lex said, well, what about polio vaccine? We went from tens of thousands of cases to very few. He said, no, that's not really been very useful either. So to me, this is very scary that someone with no scientific or medical training whatsoever, RFK Jr., could be, say, the head of CDC or Health and Human Services. This is bloody scary, folks, because you'll see they'll start eliminating vaccine exemptions, eliminating school mandates for vaccination, and we will have huge outbreaks of infectious diseases. Does it have to go to that before we realize what we're doing? So spread the word and tell people, if anything, do not vote for a person who wants to dismantle our public health system. And RFK
So,
Jr. says none of the vaccines are useful, but he's got them all. He's gotten them all. Sorry, Mark, go ahead.
and no, I was going to say that's the important question to ask these folks. And what I would say is I think I know where this stuff comes from. And a person who I really like a great deal in my personal life was in discussions with other people, and she happened to say that she doesn't know any pediatrician that agrees with the current vaccine regimen for children. Now, I don't know quite how to respond to that, and I didn't want to get into a big fight, but I think that's why it's important for people to look what the American Medical Association says about these things.
Mm-hmm.
And if you can say that they're in on some conspiracy, this is a hole that you will fall down in and never get up. You've got to be very careful. You have to stick with things that we can actually do studies on. And I think the way this happens is if someone says, is it a good idea to give a lot of vaccinations early? Someone can say, well, why not space it out? And I'll be honest with you, I haven't looked at the data. But that when you give vaccines, there are people who have studied the immune responses to them. And that's stuff that's in the published literature. And this is why the people, and again, I'm not taking a political position because I swore I would never do that. But what I will say is that there are people all over the place that want to use fears to advance their own agenda.
Mm.
And that worries me. Sometimes that agenda is putting money in their pocket. Other times it's getting on television. But the most important thing is to minimize the amount of suffering and death that you can deal with. To me, that's the most important thing. And I don't have a response to things like that, except as you say, here are a couple of people who I don't believe that either of them have ever taken a biology course at the collegiate level. And I know that was the case for previous presidents as well. So should we insist on that? Well, if you can't insist on that, you can at least insist that there be experts that advise them properly. And then people say, well, Doc Martin, what you mean is by properly is agree with you. No, the difference is there are peer-reviewed studies that can show you, we can actually find out if something is true.
Yeah.
Sometimes we don't have all the information we need. And can I add one further thing, Vincent?
Of course.
There's a problem that all of us have, you, me, everyone. Well, I won't include you, Vincent, but many people have, and I'm one of them, of what I call lottery thinking, where we don't weigh risk factors very well. So, you'll say, for example, I don't like and fill in the blank, but you do things every day that have a greater risk to them. And any type of medical procedure, any type, is going to have potential side effects and difficulties. You have to ask whether the lack of treatment has worse effects. That's what you need to ask. And people
That's
don't
correct.
do that. They tend to focus on
That's right.
this lottery thinking.
This idea that, we give too much, too many, and too much vaccines. If you had one infectious disease, you would have a greater burden of antigen, a protein, nucleic acid from that agent than you would from all the vaccines put together. And that's what you're trying to do to prevent that. Much of the rhetoric still revolves around vaccines causing autism. RFK Jr. will not back off from that. And there's been disproven many, many times. Many studies have shown that they do not cause autism, yet people don't believe it. They say the studies are flawed. They're not. There are dozens of independent studies. So I don't know how you convince people to do that. So we had a little discussion in the pre-show about this. And so let me highlight some of the comments. So Kang said, science should be fearless, but look what they did to Galileo. Brecht in his play Galileo said, I fear a country that needs heroes.
That's good.
John says, RFK Jr. in an administration is a scary prospect. It absolutely is, for sure. Okie dokie. All right, here's a question, a virus question. Can you explain the difference in the N1 protein in H5N1 versus N1 influenza's H1N1 in the 2023 vaccine, such that previous vaccination would not provide some protection against N1 TWIV 1129? My understanding is that they're distant. They're distant enough so that you wouldn't get cross protection. But if you're over 65, you may have been infected with a virus with some kind of N1 that does provide cross-protective ability to H5N1 viruses, right? Okay, there were some. Go ahead, Mark.
I was going to say, Vincent, is that I was talking to students the other day. Let me see if I can put my hand here where you can see. Yeah. So, what I say is, if my hand, you're scared there for a minute, weren't you? If my hand is an antigen, all of it. And so what I tell them is that it's all about shape. And when I used to teach intro biology, I called this issue stericity. That is to say, the molecular shape is really important. So when an immune response happens, if this is an antigen that's introduced, you might have an immunoglobulin, an antibody that binds here, or binds here. And what people forget is what antibodies actually bind to are what are called epitopes. And that's, for lack of a better term, the finest level of antigenicity that can actually bind to that portion of the antibody molecule. So what that means is you can have very minor changes in the shape of something that's able to kind of defeat a good binding site for an immunoglobulin. On the other hand, there might be changes that don't have any effect at all on that ability.
Exactly right. All right, here's a question for Mark. Does he have any general thoughts on bacterial antibiotic resistance? I'm sure you
Absolutely. And it's really an interesting subject because the first thing that I want to say about it, and I won't take too much time, is that it's not in response to the antibiotic. People tend to think that way, and it isn't. I want you to imagine that the hand of Darwin is on everything that lives. I use an expression I think Vincent likes, Darwinowing, where
Yeah.
natural selection acts on things all the time. So what I'm trying to say is that there's constant changes in shapes all the time, and they're heritable, but they're at low levels. So if it so happens that an antibiotic can have an effect on a cell, a very small change can lead to that cell being resistant to that antibiotic. Then that cell has a real advantage. Now, the second thing isn't that, it's the real problem, which is horizontal gene transfer. And this is a topic that's really important for people to understand because we're used to animals and plants mostly, and gene transfer is kind of segregated. There are exceptions, but it's really segregated. The definition of a species are things that you can and can't cross productively, right? But in the microbial world, there's a very, and I'm not trying to use this to be silly, very promiscuous exchange of genetic information. It would be like you having children with your Caesar salad at lunch. And I use that intentionally because that's the level of gene transfer that can take place. There are lots of different ways it takes place. There are small pieces of DNA called plasmids that can carry 8 separate antibiotic resistance genes on them. So a cell that receives that plasmid will become simultaneously resistant to 8 of them. If you've seen some of the work that Michael Bain has done at Harvard from the Kashoni lab where he was a postdoc, you can actually see evolution in a giant petri dish as these bacteria become progressively more resistant to a given antibiotic. But they haven't even transferred it to other things yet. So this is an uphill battle because the antibiotics don't change, but the organisms that are resistant to them can. That was long-winded, but I think you get the gist.
So the TWIV I did with the folks in Australia, two people from Europe who where phage therapy is actually more widespread than it is here. And in Belgium, for example, there have been over 100 uses of phage therapy. Um, they're much more positive about it than we are. There you go.
T evens.
Kang wants to know if certain combinations of H and N in flu viruses are found more frequently. So as far as I know, you can get all combinations. But as you know, in humans, the only three, H1N1, H2N2, and H3N2, are human influenza viruses that transmit among humans. So H5N1 does not transmit among humans. So we don't count that.
Yeah.
But all the other combinations, you can see them. You can find every combination in nature. I don't think it would be fair to count them because I'm not sure the surveillance is sufficient to get everything in every
Mm-hmm.
animal, right? So that you could say,
Mm-hmm.
is there some bias? But yes, I would think some combinations would be more fit for certain animals. For example, the hemagglutinin and neuraminidase have to match because the hemagglutinin binds to sialic acid and the neuraminidase cuts it off at the end of the cycle. So they have to be balanced. If you have a really tight binding HA and the neuraminidase is weak, that virus will be not fit whatsoever, right? So the activities have to be balanced as well. It's a good question. Okay, let's see. Let's scroll down here to another one. Mark, can you explain why there are gram-negative and positive classifications of bacteria and if one of those is worse for humans?
That's a wonderful question. And it used to be that this gram positive, gram negative was used almost like a marker. And you really want to move away from that. It's named after Christian Graham, and it's the way that these particular organisms are stained using a series of dyes, and hence the gram positive, gram negative. Basically, what you have is the cell membrane and then you have a very thick layer of peptidoglycan. And when I say peptidoglycan, I want you to think about this network that helps protect the cell against changes in osmotic pressure. So you could think of them sort of like Spanx on bacteria, or pantyhose if you prefer. But the point is, it helps protect it against Churger. So when gram positives, you have a cell membrane and then you have quite a thick layer of peptidoglycan. In gram negatives, on the other hand, you have yourself two different membranes and a thin layer of peptidoglycan, and the region between the two membranes is called the periplasm. And you might ask, why do those things exist? And I often say that's a theological question, because I don't think anyone can really know. But what I can tell you is that things with two membranes are a little bit more resistant to environmental pressure than things with one. So gram positives, and there are plenty of examples of gram positive pathogens. Corynebacterium diphtheriae is a gram positive, right? Anthrax is a gram positive. And then gram negatives, we have plenty of pathogens. Pseudomonas is that way, so, like aeruginosa and other ones. I would argue that E. coli, which isn't always a pathogen, is another one. But I do know that when the first antibiotics were being developed and they worked against diseases caused by gram positive organisms, if you wanted to use those against gram negatives, you had to modify the antibiotic to make it more permeable because of those two membranes.
So one of my guests on the TWIV last night works on endolysins, a
Mm-hmm.
protein, a phage protein that lyses bacteria, but it can't get to the peptidoglycan when it has an outer membrane. So he has to include something to hit the outer membrane with the endolysin, and then it
Mm-hmm.
will work. So you know what he said was what's his name? Can't remember. He says, looks like when you do a trial of a phage therapy, it's best to include an antibiotic with it. That works the best. In Belgium, they've had the most success when you combine antimicrobials, antibiotics with phages. Have you heard that?
I haven't. That's really
Okay.
interesting. Are
You
they
have to look
using,
up his name.
therapeutic doses of antibiotics, or is this subclinical?
It's no therapeutic. Rob Levine. Okay, here's another one. SIP3 asks, I ask how much do we know about the distances that trees and fungi are communicating? Feet, miles?
That's a really interesting question, because whole forests can be interconnected by what are called the mycorrhizae.
Hmm.
So I would probably, I'd be willing to say miles, to be honest with you.
Yeah, miles is it. Elizabeth says, I don't think it's even about believing politicians. It's more like people want to listen to people who are telling them what they already believe. Politicians are saying what people want to hear. That could
That's
be,
interesting. That's
but
interesting.
also they will vote for people who say things that they want to hear and ignoring other characteristics of those people, which is very clear in this case, right? That should be really game breakers, but they're not.
It's interesting to me, because I think one thing that helps is having people at an early age actually doing experiments. Now, I don't mean high level stuff, but phrasing a question, looking at the data, trying to determine if there's an alternate explanation, that kind
Mm,
of thing.
right,
But the
right.
way that we teach science, and I'm sorry, with all respect to teachers out there, a lot of the issues with the educational system are completely exterior to what you do. They've been imposed upon you.
Yeah.
But I think that one thing that'll help is this. When I say critical thinking, that's become code for agree with me. And that's not what I mean by critical thinking. I do find that once students have an experiment and they carry it out and they're really looking at it and thinking about it, they're so thoughtful about it. And then I tell them, you can use this same approach to anything in life. Look at the evidence. And that's hard, because we have limited time, right?
Yeah, that's it. In the end, people don't have a lot of time. So
I,
they
I would
depend.
argue.
I mean, people like us can explain things in a short amount of time, right?
I would argue, Vincent, if you're talking about epitaphs still, maybe the epitaph for the human race is TL semicolon DR.
Totally agree.
Mm-hmm.
Totally agree. Um, Here, Ripticom. Meteors and microbes in the middle of the night sky. Meteors are from somewhere. They carry something, don't they?
I think that this is a fascinating issue. There have been some recent reports that suggest now what they're doing is they're back predicting some of the first types of living things. One category of living things are called LUCA, last universal common ancestor. And what that means are organisms that use the same kinds of mechanisms, DNA, RNA, protein, replication, transcription, translation, that we do. And so turns out that things like what we're used to happened very early in Earth's history. And there are people who don't want to agree with that because they're predictions based on computer models. And I appreciate that. But I do think living things originated quite early in Earth's history. And it makes me wonder if, and Vincent will laugh out loud, I think we're all from Mars. Mars cooled before Earth did.
Hmm.
And I think life started there and was carried here by meteorites because that's certainly possible. And my guess is when we actually look for the remaining type of living things we'll find on Mars, which will be microbial, it'll be subsurface, because
Mm.
anywhere you find living, anytime you find liquid water and some kind of energy gradient, you can find life as
Yeah.
we know it.
Huh.
My
Hmm.
guess is they're going to be our relatives.
Well, we're going to go to Mars. We'll find out, right?
Mm-hmm.
Actually, we don't have to go there to find out. We could use machines, right?
Yes.
I don't know why we have to send people, but they'll just have tragedies and mess it all up. But obviously, I'm not an explorer type, at least not a traveling explorer type. I like to explore science.
You explore inside your head.
Abdur says, I understand the aversion to and luxury of avoiding public office for scientists, but Plato said the penalty for refusing to participate in politics is that you end up being governed by your inferiors. Well,
You can see why Plato was so popular at parties.
I don't look at them as my inferiors. I think they don't know science, right? And
That's right.
that they may know other things that I don't know. That's great,
Mhm.
but they don't know science, and and as someone pointed out on Twitter, but Vincent, they have to regulate science. They're lawmakers. Fine, get someone who knows what they're talking about to advise you. It's very simple, but they don't. John writes, one anti-vax talking point on the childhood vaccines is that it's too much, but you don't get exposed to more antigens in mundane events like eating, swimming in a lake. What is a good comparison? Actually, eating, you get exposed to a lot of antigens. Are you kidding? Totally. Swimming, those are all assaults. Breathing, you're inhaling tons of antigens every minute. 6 liters of air a minute go in and out of your lungs with all the stuff that's in it. However, an infection overloads you with antigens way more than any vaccination would do. Okay, so that's the best. And I already said that before. That's the best
No,
answer, I think.
and I think it's important to emphasize that people have studied this, folks. People, and when I say that, that's not a shutdown. What I'm trying to say is people have actually looked at how the immune system responds to things that happen, say, in disease or things from everyday life. We have baseline, we have experiments, and that's very different. I understand, like this. There are folks who will say things like, and I really cringe every time I hear it, natural immunity.
Right.
And there are a lot of things that are natural that aren't good for you, right? Like
Yeah,
botulinum toxin, right? So
sure.
so what I'm trying to get at here is that there are ways to actually look at the studies. I'm not recommending that everybody get a PhD. That's not what I'm saying, but there are, and this is implicit on scientists to do as we're trying here to actually explain what it is we do and how we come up with those conclusions. but I certainly appreciate the point. I think that fear mongering is probably engraved in our genes, because we don't know what's going to happen next, and that might be something bad. But we have to resist that and look at evidence where we can.
So what you're saying is we have not made the transition from hunter-gatherer, cave dweller to high-tech society.
Oh, yeah.
So we're in the cave and we're scared to death of anything, right? Because we can't
We
understand anything.
we are in the cave, but we also have iPhones.
Yeah, right. Where we can get all the information we want. Here's one for you, Mark. Have tardigrades inspired any design or material innovations?
Oh, how I love this. Yeah. The business of cryptobiosis is really interesting. And this is the resting state called a tun, TUN. As they begin
Mm.
to dry up or reduce oxygen, I think it'll also happen with temperature. They withdraw their legs and they create this resting stage. Now, remember, tardigrades are multicellular. They're not huge or anything, but they're multicellular. And what happens is they create a protein, don't ask me the name of it now, which enables them to be protected from freezing and other types of environmental insult. It gives like a glass-like chaotic folding to the proteins in a way that doesn't destroy their functioning later. And there are people studying
Mm.
that to find out how to extend the lifespan of particular proteins that you might want to use therapeutically. I've read the reports that you may have seen where they take tardigrade DNA and they put it in human cells and look for resistance to various environmental insults. That's the beginning of a Netflix special. I would love
Oh,
to star in it.
you should. You've been talking about them for years.
Yeah.
All right. Tona wants to know, when you get a blood transfusion, do you get the donor's microbes?
Oh.
I would guess so,
What's
right?
what's the bioload in blood generally
Uh,
speaking?
well, it's low unless you brush your teeth just before, right?
That's true.
Um, let's look it up. How many
That's a really
bacteria
great
in blood?
question. And your blood has all kinds of tricks to keep things from growing, which is really great, like binding iron to keep it available to the human cells, but playing keep away from possibly invading bacteria.
So according to, if you do DNA sequencing, you can estimate 1,000 bacteria per ml of blood.
Okay. And you don't know
It's
if
a lot
that's
of
alive or
bacteria.
dead.
So definitely, you don't know if they're alive or dead. Absolutely, but
Right.
you're gonna get bacteria in blood.
Hmm.
There's no question about it. But most people's microbiomes are at a high level very similar, right?
That's right.
It's only when you drill down do you see some differences.
That's a cool question.
Markle wants to know, when I get a cut gardening, should I use soap and water or peroxide? So many organisms in my organic yard where the deer, turtles, small frogs, snails, and bunnies like to eat.
I always use soap and water first, and then I hit it with peroxide. That's just me. So soap actually saponifies those outer membranes and bacteria, and so they burst. And that's why soap and water is usually the best way to reduce anything that you have on your body that you don't want that's microbial. Soap and water works very well. Peroxide's just a lot of fun.
Yeah, I think the soap and water gets rid of the bulk of everything, right?
Yeah, that's right.
And then as long as you do it for a good amount of time, right? You have to say the alphabet. Yeah, back to the blood microbes. Of course, that's why we test blood for viruses, because we don't want
Mm.
you to get viruses from blood, at least certain viruses, right? Hep C, HIV, Hep B. But there are many viruses in blood that everyone has, so we cannot throw away blood because they're there. We wouldn't have a blood supply. So for sure, you get other people's viruses when you get a blood transfusion.
So
And
I
one of the famous ones are these small single-stranded circular DNA viruses called anelloviruses. They're in everyone, and they apparently do nothing bad anyway. Okay, Mark, go ahead.
All I was going to say is when I was in college, because we were all raised in my family to give blood, because my father was a firefighter,
Hmm.
and I was cytomegalovirus negative.
Hmm.
So they really wanted my blood for preemies. Now, I don't have an unusual blood type. I'm an A positive, but I was CMV free. And then when I was in grad school, I seroconverted. I had no symptomology at all. So I was always disappointed.
Um, John wants to know if breweries are more common around schools with a good microbiology program. I think it's a
That's a fun
joke.
topic. Yeah, it's a fun topic.
Tryon wants to know what temperature does SARS-CoV-2 better infect cells? Actually, the temperature of the upper tract, which is like 33 degrees Celsius,
Mhm.
it's a little more inhibited in the lower tract at higher temperatures. Yeah.
Cool.
He wants some clinical virology news. Well, H5N1 is still limited to a few people in the U.S. and birds and cows and a few other animals. So we're not worried about it yet. Although we did a paper on TWIV showing that one of the cow H5N1 isolates can bind both alpha 2,3 and alpha 2,6 sialic acids. In other words, it already has the
Oh,
ability to bind human sialic acid. So it's a little bit scary, I think, or not scary, I would say unnerving. And Dagfinn says, at least in Norway, moonshining is more common in the areas surrounding polytechnic universities. Yes, indeed.
so in the olden days here, the honor society at my institution had something they called Mycology Fest, where they brewed beer.
Mm, that's cool. Laura writes, the majority of people follow authority, so knowledge has to come from an authoritarian or governmental source to be true. Scientists are not the same as everyone else, in a good way, of course.
Hmm.
Not sure
If
that
you were
science I'm not sure that science knowledge has to come from a governmental source. I mean, it comes from the journals, right?
right, right, um, and then but we need a better conduit to. And I hate how this sounds, but I have a piece of it here in my head. We need a better thoroughfare of knowledge from the research bench to the internet.
Mm,
We need to have ways, and that's why I've been so happy to see some papers that come out with graphical abstracts that certainly my students actually enjoy and understand. But we need to do this
yeah,
about most things in science, I think.
right, right. Tona wants you to discuss the newest on using fecal transplants for people with anaphylactic food allergies and improving their food allergic responses.
Well, I haven't heard about that. Maybe Vincent will comment on that. But what I do know is that I think where people are going with fecal transplants, and it's always fun to talk about this with 22-year-olds. I got to tell you, they really enjoy this topic. There's nothing better than poop humor. But I think the way this is going to go, where people are going to bank their own, and it's not the beginning of a joke. It's just that in the process of doing transfers between individuals, you have to go through many controls to make sure you're not transmitting a potential disease causing organism, which has happened. And then everyone freaks out. I know that fecal transplants have worked very well for the treatment of Clostridium difficile
Mm-hmm.
infections. I hadn't heard about the business of food allergies, but I love the idea because that's something that I don't remember as a child. Like, for example, the peanut allergies, and now seem to be quite common. So I don't know how
Yep.
much of that is due to changes in, the hygiene hypothesis that our immune system is trained by the microbes around us.
Well, in fact, peanut allergies and other food allergies are treatable with fecal microbiome transplants. Yes,
That's
there have been
fabulous.
a number of clinical studies ongoing that look at that, and so the idea is that the bacteria help shape your immune system, right? So, and that's what's involved in the food allergies. So that makes sense, I think. Right. So Craig has an explanation of the Greek in the apoptosis here. Oxford English Dictionary says action that the action of falling off or away, to fall off from. So that's the apoptosis definition, I believe. Doreen loves the Vincent Mark vibe. Thank you very much,
Vincent's great.
Mark. We have a good vibe.
Well, we've known each other a long time now, so
Andrew says, I finally found out what happens after death. It's a secret.
isn't that the expression, the undiscovered country? Yeah.
Nevitz Forever said, why can't you hear a pterodactyl P? Because the P is silent.
Yeah.
You've heard that one?
I was waiting for that. I knew that would happen.
Dagfin wants a Racaniello bubblehead. Well, hopefully Lise gave the link. If not, it's last week, and I'll dig it up for next week.
That's just amazing.
I don't know how much they cost. It is to be regretted that the full correction to ptomaine was not made at all at its reception into English, which would have also prevented the rise of the illiterate pronunciation ptomaine. Ptomaine, it was supposed to be ptomaine. I don't know, but he's saying it's the illiterate pronunciation is ptomaine. I wonder if you could animate it. Well, the head bobbles, right? The head bobbles. If you hit it, it does bobble. It's a bobblehead, but that's about all for the animation. I liked Mark's interview with the young black scientist whose research was advanced by his cultural knowledge, cassava.
Yeah, that was and Susan, thank you very much for that. That was a lot of fun because I knew him about 7 years ago when he was a graduate student for Rich Lenski. And it was a really fun episode because he's like become this like bodybuilder and he's super healthy and he's very inspirational. I know, and he's got such a background to him, and he's a wonderful voice for change. And I cannot think of better training than this gentleman has received. His students are going to be very lucky.
Rima would like to hear Dr. Sinusi on her HIV discovery. That would be a very interesting interview. Yeah. Mark, if you want a great New Zealand scientist, get Susie Wiles. You know Susie.
I do. Susie is really a wonderful person.
Microbiology of cheese episode was great.
Wasn't it? Yeah. My favorite is when I asked Rachel what her favorite cheese was, and yep. Her head exploded. That was a lot of fun. And then also, Hazel Barton is the cave scientist. Barton, yeah. That was amazing. Cave. Yeah.
And Dr. Geo.
Yeah, wasn't he fun?
Metallic copper versus oxides.
So I'm presuming the question is effectiveness, and this topic came up when we did the episode of Matters Microbial, and apparently, you do have to clean the copper, like you would anything else. And what I liked about it, there's a description of the superoxides that get generated, and that's kind of the explanation why it's antiviral as well as antibacterial.
Can bacteriophages infect eukaryotic cells? They're taken up, but they do not reproduce. They're certainly endocytosed, right?
Yes.
We did a paper on that in TWIM a long time ago, but they do not reproduce. All right, Mark, not. Considering Saccharomyces, do you know if there's a concrete threshold at which respiration takes over for fermentation regarding available oxygen and carbohydrates?
This takes us back to brewing. This is a wonderful question about where the cutoff is between aerobic respiration and fermentation itself. And every time that I've ever grown any type of yeast, and there's lots of different types of yeast, I've always grown them under, very, very rich media, and I've either grown them anaerobically or in the presence of lots of oxygen. But I promise you, the fermentation scientists, the Tuborg folk, for lack of a better term, know all about this.
The algae that glow blue at night are nice too. Very nice.
They do, dinoflagellates. I'd like everyone to remember that bacteria don't blink. So when you see these flashes, there's one sort of exception. Well, there are two sort of exceptions, but because bioluminescence is one of my favorite things. And the dinoflagellates are mostly what you're seeing like during the summer months, and you get that sparkling effect. It's beautiful beyond words anytime you agitate the water. And in fact, you can buy little globes with a common type of dinoflagellate in there, but you have to train it to have the right circadian rhythm and all the rest. And I'll send that link to Vincent if because they're fun to have. I've had them. But bacteria themselves are always on. Well, other than quorum sensing, but yes. They don't have and the Japanese flashlight fish actually has a shutter, so there's a light organ and it opens and closes. And that's how it's able to give that appearance. Bioluminescence is awesome.
Visto says, the mice infected with H5N1 by drinking cow milk. Yes, you can feed mice infected cow milk. They get infected. Are people drinking raw milk doing gain of function research? I don't think so. They're not modifying their genome. They're just getting infected.
So what I'm showing you here is a depiction of listeria. And listeria is really common in dairy-associated infections of non-pasteurized dairy products. So it's and I think it used to be a really major way tuberculosis got transmitted. Am I right about that? Through milk?
Yeah, that's correct.
And I also love telling people, just because it's the evening for a lot of folks, that pasteurization was not invented for milk. It was invented for wine.
Laura says mycotoxins can be dicey to deal with, though. Some can lead to disease by inhibiting various processes and enzyme pathways.
I agree 100%. There are lots of different types. The ones we're most familiar with are agricultural issues, and some of them are extremely dangerous. In fact, some of the most commonly known mycotoxins I'm aware of cause serious DNA damage. And so they can lead to liver cancer and similar sorts of things, because that's where they try to break the mycotoxin down. And this is one of the reasons I think fungal research is something that ought to be followed up on more than it is. We don't know enough.
Tryon wants to know if Candida have viruses.
Sure.
Sure. Everything has a virus, right?
That's right.
Hello from Hangzhou, China. Yu Xing. Actually, I think I met Yu Xing at ASM the last 2 years. And I think she was there at your TWIM also, if this is the same Yu Xing I'm thinking about from Baltimore, who must be visiting China. Do you know what they call the alternative medicine that's been proved to work? Medicine. Okay. And yes, being an anti-vaxxer is a money-making proposition. That's absolutely right.
I have kind of a weird view on this, because if someone has a bad idea or an incorrect idea, I'm not happy about it, but I understand where it comes from. But if you're wrong and you're saying it to make money, that's something I have real trouble with.
They do say it to make money, for sure.
Yeah.
Don't take those vaccines. Buy our product. All the top anti-vaxxers have some kind of product that they sell.
Oh, makes me so sad.
Dave H. writes, really enjoyed last week's office hours on eradication of polio on maternal antibodies. Are children born with these, or does it come through breast milk? So it passes through the placenta from the maternal into the fetal circulation. So you're born with it. But then breast milk also contains antibodies, especially the first feeding of milk. Colostrum
Colostrum.
has a lot of antibodies in it. How long does it last? No more than 6 months. It begins to decline very rapidly, and then by 6 months, it's gone.
And this is a really good example of things that we know about because they've been studied and published upon and evaluated by other scientists.
Vista wants to know if fungi have phages, and would they be a possible treatment for antibiotic-resistant fungal infection?
I think that's a really smart thing to look into. I'm not going to go chasing it down now, but what a great question for Arturo, because his interests are like mycelia; they're everywhere.
Yeah.
And this might be a really good thought. I've never heard anyone else suggest that, and it's a really good idea because every biological entity that is metabolically active, see how carefully I put that, has viruses associated with it. Vincent, what's the one you used to tell me about that we breathe in when we go to the restroom? There's one like if you sorry for this, it's graphic, but if you're using a urinal, you're putting a spray up in the air. And Vincent used to tell me you're inhaling a certain number of, and I don't know what kind of virus it was, but we don't
Oh,
know that there's any bad.
that's a it's a polyoma virus, basically. Small
Yeah.
DNA, double-stranded circular DNA viruses. Yeah, we're full of them, and we excrete them in your urine, and yes, you're in a urinal, and you inhale the next person's viruses. You should close the toilet lid when you flush. There's an aerosol. Michael Schmidt keeps his toothbrush in his room in the hotel. He never leaves it in the bathroom. He told us that months ago. Calico wants to know what type of gain of function influenza did some people do in the 2010s. Well, people have been doing gain of function with influenza viruses for decades, but what you're probably thinking about is when they took H5N1 influenza virus and adapted it to transmit through the air among ferrets. So that's a gain of function.
Right.
And that raised a lot of hackles in the world, even though it wasn't really a dangerous experiment, because in doing this and adapting it to respiratory transmission, the virus completely lost its virulence for ferrets. So, often when you tinker with a virus, you mess it up. That's the gain of function you were talking about. Rima says, we grew up on gram stain, can't get rid of it.
Rima, it was funny because I'm a terrible microscopist. And so I learned a long time ago that you can use what's called the KOH test. Put a drop of 1% KOH onto a microscope slide, pick up a colony, smear it around, it's with a toothpick, and
Mm.
lift. And if you're gram negative, you get a string. If it's gram positive, you don't. And
Mm.
it's been known since the 70s, and it isn't reliant on the phase of culture that you've got things in. And I had to convince people it was, even though it's published, I had to convince people it was a good alternative.
So Craig says fungi
Otherwise,
are
you have, go ahead.
too close to us to find antimicrobials. Is that correct, Mark?
They're awfully close to us, and that's the issue. It's the same thing with protistan infections that they have a very similar biochemistry. Now, remember, the outer surface of a fungus is generally cellulose, and so that's quite different. We don't have a lot of cellulose containing organisms in us.
We need fungi. They make antibiotics.
Some do, and some bacteria do. Absolutely.
Yeah.
And it's like saying getting rid of all bacteria. You wouldn't want to do that.
Okay, let's see here what else we have. Tom says, paraphrasing the late distinguished historian George Moss, UW Madison, a refugee from Hitler, you may avoid politics, but politics will not avoid you.
Yeah, that's true.
Tryon says, do comets only have ice?
No.
They have other things as well?
Well, a lot of them, well, they're kind of like icy gravel, but there have been a number of photochemical reactions that have taken place. You can find them, and it's not just water ice, you have ammonia ice. So you can find complex organics on comets.
Okay, John wants to know, how often do you wash denim jeans after each use? A few uses weekly or longer?
I I I wash mine after I wear them. Yes.
Steve says Dunning-Kruger, the effect, not the moderator, will be the downfall of democracy.
Yeah.
Yes, agreed. Then I just have to figure out where to go. Patricia says that's what we do with needle sticks, soap and water first.
Yeah.
Back to the question of what you do, soap and
Yeah.
water or hydrogen peroxide.
I
Does
mean,
your
hydrogen
yeah, go ahead.
I was going to say hydrogen peroxide is not magical. It certainly really disrupts the heck out of living things, including you.
Yeah,
But the thing is that that soap and water really works quick.
yep. So Nevitz wants to know, does your microbiome play a role in driving appetite? What about in driving binge eating of bad food choices or good food choices?
I think this is really likely. There's absolute connection, the vagus nerve and your gut. And
Mm,
it's really funny to think how much neural tissue you have in your gut, but you've got a lot. And there are lots and lots of microbes that are making neurotransmitters that live in your gut. And some of them use those neurotransmitters themselves. So it wouldn't surprise me in the slightest. I mean, a good example of this is there is a type of a Bacteroides, and I know that's no longer the genus name, but I'm old. And it lives off what are called sugar acids, and those are modified sugars. And what's interesting about that is they're found primarily in mucus that your epithelial cells actually make. So these bacteria induce your epithelia in your gut to make mucus because they're eating the mucus. So they are mucus farmers and they're farming you.
hmm. Did you know that there is a brewery in Kalamazoo called Apoptosis? I did not know.
Is that when you drop the beer mug?
I guess so. Yu Xing wants to know about the progress of clinical trials involving VSV vectors. I think that's a topic for TWIV. I couldn't do it right now. And anyway, we only have a few minutes left, but I will write that down and we can do that on TWIV. Absolutely. Ariozone wants to know if the hygiene hypothesis includes the impact of pesticides and now microplastics. Mm.
Wow, those are really good ideas because it's absolutely true that the microbes are kind of constantly educating the immune system. I don't know that people have looked in insects. They certainly have the effect of pesticides on the microbiome of say insects. That's been looked at a little bit. I don't know about people. It's a really interesting subject.
Mm.
Microplastics are so ubiquitous these days. I'm almost afraid to look.
Will says knowledge and scientists. You need to pick your scientists. I've been keeping a list now of experts over the pandemic who've issued dangerous information and have been
Yeah.
resistant to peer review. That's a great idea to do that. I love it.
I think the hard part in life, Will, is that you'll meet a scientist whose work you've really respected, and you'll hear them say things that are just awful, and and it's okay if it's their personal life because that's one thing, but when they start to get odd about their science, you're right. You have to put that in a bin.
Yep, absolutely. All right, Tona wants you to share a bucket list goal during your retirement.
Wow. Thank you, Tona. It's a real question what I'm going to be doing, after a couple more years. And I'll be honest with you, I wish I had a little lab to continue to do the work that I do. I enjoy it. I study bioluminescent bacteria these days, which fascinate me, and I look at variation in the amount of light that's being produced. I would love to be able to do outreach to schools. That would be a lot of fun. I love doing the podcast. I'd like to do it as long as I can, and I would like to write a book. ASM's been talking to me about it, but we'll see if they like what I write.
Sounds good. Okay, so cleaning metallic copper would mean you are removing the outer copper oxide layer, and hence it's CuO, pure metallic copper, that has the antimicrobial power.
That is my understanding.
Was it Blue Bonnet with Listeria ice cream? Ha ha ha. That's very funny. That was the contaminated ice cream, right? Blue Bonnet.
It was. Yeah.
Good job, Tona. One of the top new fungicides is produced from a fungus.
Mm-hmm.
Not surprising, right?
Yeah.
Andrew wants to know if urinals have lids in the U.S. No,
No.
I have never seen a lid on a urinal. Fisto says sampling high altitude atmosphere finds bacteria all the way to the International
Yeah.
Space Station. Earth sheds dandruff into space.
It does. And do you know what Lagrangian points are, Vincent?
Tell me.
So if you look like the Earth and the Moon, there are areas, and they're called Lagrangian points, where gravitation is balanced
Mm.
between the Earth, the Moon, and the Sun. And there appears to be dust there based on radio astronomy. I would love to see if that dust contains biological material.
Mm-hmm. Yeah. Well, how do you sample it?
Use an aerogel. I would run a probe through it. An aerogel, you know, those super light things. And this is what people used to do to collect cometary fragments, is
Mm.
they would open it up and it would get impacted in the aerogel and they would return that.
Rima says she's sick but could not skip our special guest.
Thank you so much, Rima.
And Tom says very much enjoying Mark M today. And
Thank you, Tom.
that does it for today. That has been Office Hours. I'm going to thank our moderators. We've had Tom, we've had Barb Mack, we've had Andrew, we have had Steph SF, and Les. Did I say Tom? Did I say Andrew? Yes, Les, Tom. Thank you all for moderating. Appreciate your being here. Thanks all of you for coming. Looks like 132 viewers and 133 likes. Thank you so much. Hearts to everyone. Thank you so much for liking. Thanks for coming. Mark, thank you again for being here. Really appreciate it.
I really enjoyed it, Vincent.
Next week, the 24th of July. Is that right? Next week is the
Yeah.
24th. Oh my gosh, July is almost finished. Our next live stream, office hours, will be 8 p.m. The same day, though, 1 p.m. Eastern, we'll have a TWEVO live stream with Nels LD. Okay, so next Wednesday, 2 live streams from MicrobeTV. So we'll see you in about 6 months here again, Mark.
All right.
All right, everybody. Thanks, everyone, for coming. We really appreciate your being here. And until next week, be safe. And let me find the plaque logo. Good night, everyone.